chain. This is, after all, the essence of transcription, the fidelity of the process. It is the essential requirement for the accurate readout of the genetic information. The answer to that question came only some years after the structure was derived. And I will preface it. So first just show you a slide to orient you further. So in the next slide, you see the same thing that I show you here, except in a space filling or a surface representation, rather than a ribbon or chain trace of the protein and DNA. So here it is, the same as in the previous slide, only in a surface representation. And what you see in gray are the cut surfaces from which the protein was removed, as I told you in presenting the previous slide, to expose the path of the DNA and RNA. But what you can further appreciate from this slide, there is a hole that passes all the way through the enzyme, from the bottom to the top. It is through this channel that the nucleoside triphosphates, the substrates for the formation of the RNA, enter. They first contact the enzyme at a so-called E or entry site. All four nucleoside triphosphates, AU, G, and C, will bond with equal affinity to the entry site. Only the one of the four that is correctly matched to the coding base in the DNA will rotate into the so-called A or addition site. And now I will show you a structure with such a correctly matched nucleotide bound in the active site prior to its addition to the growing RNA chain. In this next slide, and several of those follow, we omit almost all of the protein and focus, zoom in, as it were, on the region around the active center. So here is what I have just said, zooming in on the active center. Most of the protein is gone. Here is the template DNA directing RNA synthesis. Here is the RNA of the hybrid helix with the DNA. Here is the nucleoside triphosphate seen in the structure, residing in the active center before addition to the growing RNA chain. What we discovered about 5 or 6 years ago was an additional protein element, a component of the RNA polymerase that had not been seen earlier because of its degree of mobility. It is referred to as the trigger loop. In all structures that we determine in the absence of correctly matched nucleoside triphosphate in the addition site, the trigger loop was far distant from the active center, and in one case even disordered, as indicated by the dashed line. Only in the example with a correctly matched nucleoside paired the coding base in the DNA was the trigger loop in a position adjacent and able to interact in the manner that is emphasized here. So the trigger loop now is purple, this is the protein, and the dashed yellow lines indicate its interactions with the base of the entering nucleoside triphosphate, the second of the three phosphates, the so-called beta phosphate of the nucleoside triphosphate, and indirectly, not shown here, also interactions through the polymerase with the sugar moiety of the nucleoside triphosphate. In consequence of the precise alignment to better than 1 angstrom precision, a side chain of a histidine residue of the trigger loop is in a position to promote the flow of electrons during attack of the 3' hydroxyl group of the growing RNA chain upon the alpha phosphate of the triphosphate and displacement of the beta and gamma phosphate due to then phospholipid hydride bond breakage. So the trigger loop then couples recognition of the correct nucleoside triphosphate to catalysis and RNA chain growth, to the formation of a phosphodiester bond and addition of one nucleotide to the growing messenger RNA. Now I will turn from the detailed mechanism of transcription to what I alluded to before concerning the general transcription factors, the recognition of the start site of the gene and the beginning of the transcription process. Here again, I will show you only one example from the work done over the years that is merely illustrative. I'll begin with the smallest and simplest of the 5 general transcription factors, so-called factor B. From what I tell you about the information gained from studies of just this molecule and its interaction with RNA polymerase, I believe you'll get a sense or flavor of what can be learned from structural study of the entire transcription machinery. The next slide shows the result from structure determination of a complex of the RNA polymerase enzyme with factor B. Here is the polymerase again. This is the same view that I showed you originally, a view from the top. The cleft through which the DNA enters is where I indicate by the pointer. The active center would be about where I show you here. The clamp in gold, the wall in blue. And in this structure of a complex with factor B, red shows the actual chain trace, a ribbon diagram of factor B. Now, separately, some years ago, approximately almost 40 years ago, Stephen Burley, the late Paul Sigler, and their colleagues determined the structure of a fragment of factor B in a complex with a fragment of one subunit of factor D, which is called Tautobox binding protein, and DNA containing the sequence TATA, the top sequence. Their structure is shown here. In this small 3-component structure, the factor B is red, just as in what I have presented a moment ago. The fragment of the Tautobox binding protein is purple, and the DNA, as in the previous slides, is blue and green. What Burley and Sigler noted, the DNA is sharply bent in this structure. The arrows indicate the directions of the axes of the double helix of this fragment of DNA. Now, if you look closely, you may notice that the alpha helices of factor B, which is in red, in the old Burley-Sigler structure, match the alpha helices in red that I point to here of factor B in our crystal of a complex with RNA polymerase II. Indeed, the factor B here can be perfectly superimposed on the one from our more recent structure. And so this entire complex can be aligned, can be docked to the structure, and that is done in the manner shown here. What we do next is an exercise in model building. We simply extend the ends of the fragment of DNA in the Burley-Sigler structure with standard B-form DNA molecules. So there's nothing complicated. We just add a bit of the well-known DNA structure to the ends. And in this way, we arrive at a model of what we call the closed promoter complex, which occurs prior to the initiation of transcription. We call it a closed promoter complex because it is entirely double-stranded. There is no unwinding of the promoter DNA, such as I told you is required for and is observed in the active center of molecules engaged in transcription. Now, we know from many studies done over the years that unwinding begins at the start of transcription at a point approximately 20 base pairs from the Tautobox. So at position approximately -11 if the Tautobox is at -30. That point in our model of a closed promoter complex lies directly above the active center, where you will recall there is a catalytic magnesium ion in the structure. Now, I will extend this exercise in hypothesis or in modeling just one step further. and go from the closed complex to the open one in which transcription actually begins. In order to do that, I'll go back to a ribbon diagram, and I'll rotate it around by 90 degrees, and I do that for the purpose of comparison with the structure of the actively transcribing RNA polymerase that I showed you at the beginning. So here is that structure, and it only differs slightly in orientation. In this original picture that I showed, the DNA was entering from the right. Here the double helix enters from the left. But the orientation is such that I can superimpose the protein here on the one on the left to produce this picture. And what you see is that the upstream end of the RNA-DNA hybrid helix in the transcribing complex lies directly beneath the point where unwinding begins in the closed promoter complex model. So to pass from closed to open, all we need to do is remove this portion of the DNA and replace it by what we see in the actively transcribing complex, and also join the broken end here to the one from the transcribing complex. And so that's done in this next slide, and that simulates, as it were, the transition from a closed to an open promoter complex. It is at this point that transcription begins. I could tell you in much more detail about how that takes place, but there won't be time. I could explain to you how, in 2 stages, the polymerase, having initiated transcription, then disengages and escapes from the promoter to transcribe the body of the gene. But again, there won't be time during the course of the lecture. I'd be happy to tell you and answer to questions either here in the hall or probably later. Now, what I'd like to do is present a series of slides just to give you a sense of how we bring into the picture not only the C-terminal portion of factor B that I have mentioned and the promoter DNA, but all the rest of the proteins of the machinery. Now, what I will illustrate with these slides is only an approximate view, but it serves for the purpose. It's actually what we knew as of about 3 years ago, and I'll indicate briefly how what I present now is in the process of being replaced by more detailed information. So we begin with a picture of the polymerase in the center. This is the view that is familiar to you. It is a view of the enzyme from the top. The DNA, as I told you, enters through this channel from the left. Here's the structure of the portion of factor B that I have mentioned, and then, as I said, the promoter DNA and the cotton box binding protein. Didn't have time to tell you about the rest of factor B, which is in yellow, or the structure of the rest of the molecules. But I will simply include them here for the purposes of illustration. I told you that we originally solved the structure of the polymerase lacking the 4th and 7th larval subunits. They could be added by means of structures determined later. They interact at this point on the surface. Factor F, we know from primarily from molecular microscope studies, is situated in the manner shown approximately here. The amino terminal portion of B, again, I had to omit the discussion for the reasons of just the time of the lecture. Here is what I just explained about the way the C-terminal part of B brings the promoter into the picture and places it in an appropriate location on the polymerase for interaction with the start site of transcription. Factor E interacts with H. It brings H to the polymerase surface in the manner illustrated, and in this way we arrive at a picture of the complete transcription machinery, lacking only the medium of transcriptional regulation that I will come to in just a moment. Now, as I said, this was a picture that we could produce based on information we had a few years ago. We will shortly have a more precise and a definitive